This system will focus on muscles. Each muscle is considered to be a separate organ. The functions of muscles are movement, stability, heat production, control of body openings and pathways, and glycemic control, or control of blood sugar. As you may see from this list, muscles are highly active. They have a lot of energy requirements and they produce a lot of waste. Muscles are highly vascular. As a system, the muscular system is primarily regulated by the nervous system. Individual muscle cells must be stimulated in order to contract. As previously mentioned, there are three types of muscle tissue. Skeletal muscle is the most common. It's voluntary, the cells are striated and multinucleated, and skeletal muscle acts on bones. Cardiac muscle is found only in the heart. It's highly active, cells are striated. Cardiac muscle is considered to be involuntary, and the cells are connected by structures called intercalated discs. Smooth muscle is involuntary and it's not striated. Smooth muscle is found in digestive system tissues, for example, respiratory system tissues, cardiovascular vessels, etc. All three types of muscle tissue have the following characteristics. Number one, excitability. Muscles respond to electrical signals or stimuli. Number two, conductivity. Muscles have the ability to carry electrical signals. Number three, contractility. Cells have the ability to shorten, which will pull on bones. Number four, extensibility. Muscle cells have the ability to stretch past their initial length. Number five, elasticity. Muscles have the ability to return to their original shape. Skeletal muscles come in a few different shapes. Muscle strength is determined by the shape and the direction of the pull. Shape is determined by the orientation of the muscle cells within the muscle. Fusiform muscles are wide in the middle and have tapered ends. Parallel muscles are uniform in width and parallel cells. They look like elongated straps and can span long distances. These things shorten more than other types, but produce less force due to fewer cells. Triangular muscles are fan-shaped, they're broad at one end and narrow at the other. They are relatively strong due to the high concentration of cells. Pennate muscles are feather-shaped and generate the most force. Circular muscles are round and usually control openings and are sometimes known as sphincters. Muscle compartments are a group of functionally related muscles enclosed in connective tissue. Muscles and muscle compartments attach to bones both directly and indirectly. In a direct attachment, the gap between the muscle and the bone is so small that it looks like the muscle attaches directly to the bone. In actuality, the muscle is attached to bone by little collagen fibers which are continuous with the periosteum on the bone. In an indirect attachment, it looks like the muscle stops before it reaches the bone. The gap between the muscle and the bone is spanned by tendons. So in indirect attachments, tendons act as the connector between the two structures. Muscle works on bone in much the same way that we use levers to lift things. With levers, one end of the lever needs to be stationary while the other end will do the moving. For muscles, one end of the muscle will attach to the bone that will remain stationary, while the other end will be attached to the moving portion. The end that attaches to the stationary bone is referred to as the origin. The muscle end that attaches to the bone that moves is referred to as the insertion. For instance, your textbook gives examples of the biceps brachii. The origin for the biceps brachii is on the scapula, and the insertion is on the radius. When the muscle contracts, it acts on the radius and brings about flexion of the elbow. A muscle action is the effect produced by a muscle. Muscles don't work in isolation. When we have an action, there is always a prime mover, a synergist, an antagonist, and a fixator. The prime mover produces the primary force for the action. The synergists aid the primary, and the fixator holds the bone in place. When we have an action, we always need a counteraction to limit excessive movement and speed. The antagonist provides this counteraction and opposes the prime mover. In other words, antagonists work to keep in check and balance the actions of the prime movers. The antagonist will keep the prime mover from moving too far and too fast, thus causing injury to the joint. Muscle structure. As we look from the outside to the inside, muscle cells have numerous different structures. The entire muscle is composed of a bundle of fascicles. Fascicles themselves are bundles of individual muscle fibers or muscle cells, and the muscle fibers, or the muscle cells, the key players in the group, are long fiber-like cells. Each structural feature is surrounded by separate connective tissue. The connective tissues are as follows, working from the outside to the inside. Number one, the fascia or the fascia is the connective tissue that separates muscles from other muscles or separates muscles from the skin. The epimysium is the connective tissue that surrounds the entire muscle. The perimysium is the connective tissue that surrounds and separates individual fascicles. And the endomysium surrounds the individual cells which are found inside the fascicles. Here are some helpful terms. We will often refer to epi and peri and endo as prefixes. When you see these, epi, E-P-I, always means outer or near the surface. Endo, E-N-D-O, always refers to inside or in the center, and peri, P-E-R-I, always refers to around or the perimeter. The suffix or ending mysium refers to muscle. This is how connective tissues get their names. The prefixes will tell you which layer you're dealing with, and the suffix will tell you about the system that you're involved in. Muscle fibers contain many of the same organelles found in other cells, but because of their functions, we give them special names. The sarcolemma is the name for the muscle cell membrane. Transverse tubules, or T-tubules, are unique to muscle cells. They are inward folds of the sarcoplasm that run through the cell. The sarcoplasm is the name for the cytoplasm of a muscle cell, or the contents. The sarcoplasmic reticulum is the smooth muscle within the muscle fiber. In muscles, we also find large amounts of a red pigment called myoglobin. Myoglobin works to store oxygen. Muscles require a large amount of adenosine triphosphate in order to fulfill their functions. In order to produce ATP more efficiently, muscles must have oxygen. Muscle cells also require a large supply of glucose or sugar in order to produce the ATP. Muscles usually do have quite a bit of stored glycogen, the polysaccharide made up of individual glucose molecules found within the sarcoplasm. You can consider this to be stored potential energy. We will discuss the production of ATP in future slides, but it's helpful to remember myoglobin and glycogen. Finally, myofibrils in the muscle cell are long, thin bundles of protein microfilaments. There are about 11 or 12 myofibrils per muscle cell. Each myofibril is made up of 3 types of myofilaments. Learn more about the myofilaments on the following slides. This slide will help you review the structures that you'll be responsible for related to a muscle cell. Myofilaments are thread-like complexes of hundreds of protein molecules. There are 3 basic types. Thick filaments are also known as myosin. They are made of a protein called myosin that has little golf club-shaped head structures that face outwards from the bundle. Thin filaments are also known as actin. They are made of a protein called actin, and these will be the threads to which myosin heads bind. Also found on actin is troponin and tropomyosin. Tropomyosin is a ribbon-like protein that wraps around actin and actually blocks the binding sites, thus preventing myosin from attaching. Another protein called troponin binds to tropomyosin and is also able to bind with calcium. Together, troponin and tropomyosin cover the binding sites on actin. When calcium binds to troponin, it allows tropomyosin to be pulled away from the binding site on actin, and then the myosin head can bind. The third type of filaments are small and made from elastic called titin. Myofilaments are thread-like complex of hundreds of protein molecules. There are 3 basic types of myofilaments, in a muscle cell. Number 1. Thick filaments are also known as myosin molecules. They are made of a protein called myosin, and these have golf club shaped heads that face away from the bundle. Number 2. Thin filaments are also known as actin. These are made from a protein called actin, and these are the proteins to which the myosin golf club shaped heads will bind during contraction. Related to actin, there are 2 additional proteins. Tropomyosin is a ribbon-like protein that wraps around actin and blocks the binding sites on actin for myosin. Another protein related to actin is called troponin. Troponin binds to tropomyosin, but is also able to bind to calcium. Together, troponin and tropomyosin cover the binding sites on actin. When calcium is present in the sarcoplasm, calcium binds to troponin and causes tropomyosin to be pulled away from the myosin binding sites on actin. The third type of protein, or myofilament, in the muscle cell are called elastin. Elastin molecules are made of titin, a springy protein that helps anchor myosin and stabilize it to prevent overstretching. At the top of this diagram, there's a single myosin molecule. Several hundred of these to combine to form the total thick filament. The next molecule with the red beads represents actin, or the thin filament. The white ribbon protein that wraps around actin is called tropomyosin. Attached to the tropomyosin is the final molecule called troponin. Again, this complex blocks the binding sites on actin so that myosin cannot normally bind. When calcium is present, only then will the troponin and tropomyosin complex move and allow the thick filaments to interact with the thin filaments. As you work down a myofibril, the myofilaments are arranged in unique patterns. A Z-line or Z-disc breaks up the myofibril into small segments and provides a place for elastin and actin filaments to bind. Each end of a myosin is anchored to an elastin filament that overlaps with actin. The overlapping shows up as a dark band called the A band, and this will be the striation that you see when you look at this type of tissue under slides. The organization of myofilaments from Z-disc to Z-disc is called the sarcomere. The sarcomere is the functional unit of muscle contraction. We'll now have a chance to examine a little bit on how the nervous system interacts with other systems, in particular the muscular system. A synapse is a functional connection between a nerve and its innervated cell. In this case, it will be a muscle cell. This is where the signal is transferred from the nerve cell to the muscle fiber. The specific synapse between a nerve and a muscle fiber is called a neuromuscular junction. The point of connection on the muscle fiber is specifically referred to as the motor end plate. The following structures are part of every neuromuscular junction. There's always a nerve fiber, which will bring information to this sequence. The synaptic knob is the end of the neuron or the nerve cell that stimulates the next cell. Inside the synaptic knob are synaptic vesicles, small little bubbles that store chemicals called neurotransmitters. In this case, the neurotransmitter stored in the vesicles of this motor neuron will be acetylcholine or ACH. The synaptic cleft is the space between the synaptic knob and the motor end plate. On the surface of the muscle cell, there will be ACH receptors or acetylcholine receptors. Within the space, there is an enzyme called acetylcholinesterase or ACHase. This enzyme will break down ACH or the neurotransmitter after it has been used to stimulate the muscle fiber. Review the structures that you've previously learned on this slide. A motor unit consists of the motor neuron and the muscle cells that it innervates. This picture, 2 motor units are shown, one in red and one in blue. Remember that a motor unit is one neuron and all of the muscle fibers that will be stimulated by that fiber. One neuron branches multiple times to stimulate more than one muscle fiber. Muscle fibers are able to contract as a unit because the nerve branches stimulate each fiber simultaneously. This slide shows the neuromuscular junction in relation to the T tubules and the sarcoplasmic reticulum of the muscle fiber. Muscle contraction occurs in several steps. First, a nerve impulse is sent down a nerve fiber and it arrives at the neuromuscular junction. After the impulse arrives at the synaptic knob, the knob allows calcium to enter from the extracellular fluid. This extracellular calcium, as it enters the knob, stimulates the vesicles within the knob to release the neurotransmitters, or ACH, via the process of exocytosis into the synaptic cleft. As ACH leaves the synaptic knob and diffuses across the synaptic cleft, ACH binds to receptors on the sarcolemma of the muscle fiber. The binding of ACH causes a brand new action potential to occur on the sarcolemma. This action potential spreads in a wave-like fashion along the sarcolemma and then gets transported down T-tubules. Impulses traveling down the T-tubules travel very close to the sarcoplasmic reticulum. Impulses in the T-tubules cause the sarcoplasmic reticulum to release calcium into the sarcoplasm of the muscle cell. Once calcium is released, it's possible for the proteins within the sarcomeres to interact. This portion of the process, focusing on the filaments, is specifically called the sliding filament theory. Calcium from the sarcoplasmic reticulum binds to troponin, which causes the troponin and tropomyosin complex to shift. This will expose myosin binding sites on actin. ATP is broken down into ADP and phosphate to provide the energy for the myosin head in this process. This is done with the aid of an enzyme called ATPase, or adenosine triphosphatase. Once energized, myosin binds to the actin and forms a crossbridge or connection between the thin and the thick filament. Once the crossbridge is formed, the myosin can then shift its head and pull the actin forward or towards the center of the sarcomere. This is referred to as the power stroke. After the power stroke, myosin releases and binds to another ATP, and the process continues. When the myosin releases to form another crossbridge, actin does not slide back into its original position because there are numerous myosin heads at different portions on the molecule. So while some of these are releasing and reshifting, others are forming and pulling. Muscle contraction is an involved process, and it may take you a few times going over the steps before they sink in. One of the ways to learn the steps is to simply write them over and over again. This will help you compartmentalize them. If you click on this link, this will also take you to an animation, which is sometimes useful. Also, in your text, there are pages that do a great job of linking this content with what happens actually on the pictures. Once the muscle contracts, it must then relax in order to be used again. There are several steps involved in muscle relaxation. Number one, the nerve impulse stops. This will result in calcium no longer entering the synaptic knob, and if calcium no longer enters the synaptic knob, neurotransmitters won't be released. Acetylcholine in the synaptic cleft separates from the receptors on the sarcolemma and is broken down by the enzyme called acetylcholinesterase. The action potential on the sarcolemma ceases, which means there will be no electrical current traveling down T-tubules. Calcium in the sarcoplasm is then pumped back into the sarcoplasmic reticulum. Calcium leaves troponin, and thus the troponin and tropomyosin complex will return to the original shape, blocking the actin and myosin binding site. When this happens, everything slides back into its original position and the muscle returns to its resting state. The energy needed for muscle contraction comes in the form of ATP, or adenosine triphosphate. ATP stores its energy in its various phosphate bonds. In order for the energy to be released from the bonds, a decomposition reaction must occur. Adenosine triphosphate is broken down into adenosine diphosphate plus a free phosphate, and energy is released. Please remember that this reaction is facilitated by an enzyme known as adenosine triphosphatase, or ATPase. Oftentimes muscles have stored ATP, but they most commonly must build ATP. Glucose is used as the primary fuel. If oxygen is present, we refer to the processes as aerobic. If oxygen is absent, we refer to the processes as anaerobic. Both processes start out with a common step known as glycolysis. This is a great mechanism that's used to convert one glucose into 2 molecules of pyruvic acid. This diagram illustrates glycolysis as well as anaerobic fermentation and aerobic respiration. Glycolysis is the first step in ATP production. Glucose is used by a cell to add 2 phosphate groups to 2 molecules of adenosine diphosphate, or ADP. Each molecule of ADP receives a phosphate. As a result of these steps, glucose is converted into pyruvic acid. What happens to pyruvic acid will depend on whether oxygen is available or not. If oxygen is not present in the system, the process continues as anaerobic fermentation. During this phase, glucose is converted to lactic acid. Lactic is the substance that makes you feel sore after muscles have been working. Because 2 molecules of ATP were actually produced, it's possible to say that anaerobic fermentation does result in the production of ATP. If oxygen is present in the cell where these steps are taking place, aerobic respiration occurs. Oxygen is used to convert pyruvic acid ultimately through a series of steps that results into the production of a large amount of adenosine triphosphate. Additionally, water, heat, and carbon dioxide are byproducts of this pathway. Both systems utilize glucose, but aerobic fermentation results in only the production of 2 molecules of ATP per glucose. All things considered, and in the best possible scenario, with oxygen, 36 molecules of ATP can be produced from the similar glucose if oxygen is around. Why doesn't the body use oxygen all the time? Well, the system takes a little bit of time to get going. It usually kicks in after about 40 seconds of continuous work. It is possible for this to continue for quite some time if oxygen is available. During exercise that lasts over 10 minutes, about 90% of the ATP available and used for that activity will come from this system. Also, the more you rely on the system, the more efficient that it becomes. If it's used extensively, a small amount of lactic acid can accumulate. It also requires a good supply of glucose, water, and minerals to make this thing process. So to summarize, pyruvic acid is produced as a result of glycolysis. What happens to pyruvic acid will depend upon oxygen presence or absence. If oxygen is not available, pyruvic acid is converted into lactic acid with a net gain of 2 molecules of adenosine triphosphate. If, on the other hand, oxygen is present, the system will continue and the net yield is approximately 36 molecules of adenosine triphosphate. Our muscle cells are very efficient at finding energy sources to meet their immediate needs. In a muscle cell, usually there is quite a bit of glycogen and myoglobin, as these become readily available for ATP production. In general, energy usage can be broken down into 3 types: immediate, short-term, and long-term. Immediate usage often utilizes whatever is currently available in the muscle cell. One system in particular, the phosphagen system, is essential for providing immediate ATP. This system is explained on the next slide. Shorter-term use allows us to produce ATP while we are waiting for the respiratory system to increase oxygen availability. This system is called the glycogen lactic acid system. We saw how this system utilizes ATP in the previous slides when we talked about anaerobic fermentation. And finally, Long-term ATP usage or energy availability occurs when our respiratory system is able to supply sufficient oxygen to cells that are using glucose for this process. In this stage, we primarily use aerobic respiration, and as we've seen, the payoff in ATP is much greater. The phosphagen system does not require oxygen, so it's considered to be anaerobic. It provides maximum strength and speed, but only for 6 to 10 seconds. This is a very small supply of energy, but it can be used quickly. For example, if you have to jump out of the way of the bus, this is the energy that will be available to you. It's also used for short-duration athletic activities like 100-meter dash or swinging a baseball bat, for example. The top part of this diagram, myokinase, is the enzyme that's used to transfer one phosphate from an adenosine diphosphate molecule to another adenosine diphosphate molecule. In the bottom portion of this diagram, creatine kinase is the enzyme that acts as the transfer molecule, and it helps a phosphate move from one molecule of creatine phosphate to a molecule of adenosine diphosphate. Both energies act like postal carriers. You drop the letter in the mail, and the postal service delivers the mail to the intended target. This diagram is useful in demonstrating how the different systems work with each other. If you use aerobic respiration for long enough, for example, you'll give the phosphagen system time to recover, and there will be a short, limited supply of oxygen available. This is why long-distance runners, for example, can have a sprint at the end of the race, as their phosphagen system is able to kick in. It's possible to use muscles past the point at which they work effectively, especially if they have not been trained or if you're asking them to do a large amount of work. Muscle fatigue can occur in a number of ways. For example, it's possible to run out of the necessary electrolytes or energy supplies and molecules, or it's possible to be fatigued because of a buildup of lactic acid. If we don't have the proper molecules to fuel the process, the muscles simply won't be able to work. When lactic acid builds up in a muscle cell, it lowers the pH and allows the muscle cell to work, although less efficiently. For example, it inhibits enzymes that are needed for contraction. Additionally, it's possible for muscle fatigue to occur because the motor nerve fibers run out of neurotransmitters. If ACH is limited, the nervous system can no longer signal muscles. Quick note about lactic acid. It's possible for lactic acid to be recycled. When lactic acid is produced, it's released from muscle fibers and it travels to the liver, where it can be converted back into glucose. So if you work sore or fatigued muscles easily, or if you get a massage, or if you engage in stretching, it's possible to increase blood flow and increase the rate of lactic acid conversion back into glucose. The strength of a muscle, or the force that it can generate, is influenced by a number of factors. For starters, the shape of the muscle cell is largely important. Pennate muscles are stronger, for example, than parallel muscles. This is due to how the fascicles are arranged within a particular muscle. Also, in general, thicker muscles have the ability to produce greater forces than thinner muscles because of more actin and myosin cross bridges. The size and the number of active motor units also impacts the force that's generated. The larger the motor unit, the stronger the contraction. Additionally, the more motor units that are activated, the stronger the contractions will be. The force of contraction is also influenced by how frequently the stimuli are sent from the nervous system to the muscle. The quicker and more frequently the stimuli are sent, the stronger the contraction will be. As mentioned earlier, fatigue can also play a role in the strength of contraction. Muscle can gain endurance through aerobic exercises. Exercises such as jogging and swimming can cause your muscles to become more efficient at using oxygen, and they can increase the amount of oxygen that's available for uptake. We can increase the number of myoglobin and mitochondria in a cell by constantly using them or subjecting them to aerobic or anaerobic stress. These types of exercises also impact how your cardiovascular system, your respiratory system, and your nervous system work in conjunction with your muscular system. When we see muscles begin to gain in size as a result of work, we get a sense of growth and satisfaction. When our muscles work, what's actually happening is that the cells are increasing their protein content. We're not actually causing new muscle cells or new muscles to develop. We're simply increasing the protein content in the available cells. There are several ways in which a muscle can contract. Isometric contractions occur when a muscle generates force, but there is no change in muscle length. These types of contractions result in no movement between the bones involved. For example, if you are standing still or holding a yoga posture, your muscles are working, but there is no change in length. Thus, there will be no movement. Isotonic contractions occur when muscles generate force and pull on bones, and movement results. We use isotonic contractions to sign our name, to walk, and to type. Furthermore, isotonic contractions, those involving movement, come in a couple of varieties. Concentric isotonic contractions occur when a muscle shortens and usually brings about flexion at a joint. Eccentric contractions of the isotonic variety occur when muscles lengthen to control the return or the extension at a joint. Cardiac muscle, the muscle found in your heart, is striated and has intercalated discs that connect cells to adjacent cells. Within the intercalated discs, there are gap junctions which allow ions to move from the cytoplasm of one cell to the cytoplasm of another cell. Technically, cardiac muscle does not need to be stimulated by the nervous system because it's autorhythmic. The cells have the ability to generate their own action potentials. We do, however, use our nervous system to increase or decrease our heart rate depending upon what we need. Cardiac muscle has a lot of available glycogen and myoglobin and is almost exclusively aerobic. Furthermore, for the most part, we don't have voluntary control over our cardiac muscle. It's possible using meditation and biofeedback to gain control or to be able to influence cardiac muscle slightly, but for the most part, it's controlled by our autonomic nervous system. Smooth muscle tissue contains cells that are fusiform in shape and can be arranged in several different fashions. Although there are actin and myosin molecules in smooth muscle cells that facilitate contraction, the actin and myosin are not arranged in the same way that they are in cardiac muscle or skeletal muscle. Therefore, we consider smooth muscle to be non-striated.